Lions Rock Productions.
This is Popping the Bubble with hosts Sandra Ponce de Leon and Pete A. Turner.
Hi, my name is John Butler. I'm the founder and CEO of QuantumCyte, and you're listening to Popping the Bubble.
Hey, John. So great to meet you and to have you on the show. I know you're doing something very, very important because you're improving cancer outcomes. I have no idea exactly how you're doing that. What I'm reading is very challenging to understand as a non-scientific person. Before we get there into what QuantumCyte is about, maybe we could talk a little bit about your journey. You definitely have a lot of experience on the medical and science side and developing new patents. So I'd love to just hear a little bit more about your background.
Okay.
What led you here?
I'll start from the beginning.
Oh, the beginning, yes.
Yeah. So I started my career biotech in the Silicon Valley, working at a company called Affymax Research Institute, where I was doing basic research, drug discovery. And during that time, I found that I had a knack for building tools for answering complex biological questions. Really enjoyed it. Not an engineer, I'm a scientist by training. I have a lot of friends that are engineers, and so we would look at these complex problems and say, let's build a technology around this that will allow us and researchers and clinicians to answer these questions more effectively or more straightforward. I continued my career after Affymax. I went to a company called Applied Biosystems, where I was in manufacturing and operations. I was running the process development group for a lot of their DNA-based products, and then moved on to a company called Pacific Biosciences that was developing a very cool high-tech DNA sequencer. And that's and I was running their manufacturing facility, and that's where I met our co-founder Bidhan, who was was in development. And we worked very closely together, joined at the hip for 4 or 5 years, and moving these very complex products and processes from development into manufacturing and then out the door. So we're very commercial guys, and we can commercialize these products. How I got involved in QuantumCyte, decided to start the company, was while I was at Pacific Biosciences, my wife Stephanie was diagnosed with late-stage metastatic cancer.
Wow.
It's called carcinoid cancer. It's a rare, incurable, untreatable disease.
Oh, my gosh.
And there are no drug candidates available to extend her life. I'm happy to say that after 10 years, she's been progression-free, and so she's in good shape.
Right.
So
Wow.
that's great.
Yeah.
At the time, she was diagnosed, knew nothing about cancer. I was going down this commercialization path in my career. After about 2 years of figuring out how to manage what's called our new normal, I decided to figure out what was going on inside her.
The new normal being 1 in 3 people are predicted to get cancer in their lifetime?
No, the new normal is your life. You define your life as before cancer and after cancer, right? A lot of doctors, a lot of operations and so forth, and managing our kids and so forth. After 2 years, we figured out what that new normal was and had a better idea of sort of how she was going to do, but not that clear. And so I decided I need to learn about cancer.
Wow.
Right. I called people in my network. Bidhan was one of them. I know a lot of people that do fantastic things in computational biology and oncology, and a lot of people that do DNA sequencing. And that's kind of what I have done all my life. Said, you know what, the current technologies in the market aren't going to help us. So the goal is to find a drug for my wife.
Yes.
Right? With little to no scientific information. And so we stumbled onto actually an article in Science called Cancer's Circulating Problem, where they talk about these cells that are floating in the circulation of late-stage cancer patients. They're called circulating tumor cells. And I said, wait a second, if I can take her cells and throw drugs at them and measure a response, I might be able to find a drug for her. Right? And I look at the whole cell. And we proposed that idea about 7 years ago, to take these cells that are floating around in the blood. And at that time, circulating tumor cells were not, the value or the prognostic value was unclear. So when we proposed that to sort of the general cancer community, we got pushback. Well, fortunately, we live close to Stanford. So I went to the lab of an individual named Ron Davis, who's a luminary in the field of genetics, builds DNA sequencing technologies and so forth. And I proposed this idea. And he said, you're way ahead of the curve, but you need a place to incubate. And so he brought me into his lab as what's called a visiting scholar.
Wow.
So I was in his lab for 2 years, incubating. What we decided as a team, myself, Bidhan, and a couple other people, is the only way we could solve this problem or do this is to build a new technology. It took us a couple of years to realize we build technologies let's go build a technology to do this.
Right. And how is this approach different than what's currently used to treat this particular type of cancer?
So this particular type of cancer, there are no treatments.
There's no treatments.
Yeah. A good friend of mine, his name is Noel Warner, told me when I first started and said, I want to find a drug for my wife. He said, you got to get the cells. You have to test the cells. And so we had to figure out a way to get the cells and then build a technology around that to go and do this. Now, that was our original idea. What we're currently working on at QuantumCyte, and this is through the IndieBio program, we came in with this idea of doing this, what's called single cell analysis. And we had this idea, we're going to go do what I just described to you. And through the IndieBio program, we found that our technology could be used in a bigger market and could be used for more, what am I trying to say here?
Beyond cancer.
Beyond cancer.
Wow.
But also we could help a lot of other cancer patients, because our goal as a company is to help cancer patients extend their lives, and specifically late-stage metastatic cancer patients.
Incredible.
That's our goal.
Wow.
That's, yeah. I don't want to put you on the spot to make you predict the future, but let's talk about the future. When does this happen? I mean, there's people that right now are like, this could change the game for me, and then that leads to, I'm willing to be tested, try it on me. And that also brings up, problems that you have to deal with. But talk a little bit about that. Like, how does that impact what you guys are doing? What do you see?
We truly see our technology as transforming the way that patients get information on how they would respond to a drug or what's going on with their cancer. Any cancer patient, we're cancer agnostic. From our perspective, this will start. We will start pushing hard on this once we receive funding, and we're right in the starting gates. Right, we've got all of the talent we need. We have the technology. We have the data that says it'll work. Now it's just a matter of getting the capital to go fast.
What was that moment like when you said you have the data that says it's going to work? So what was that moment like when you had that?
Yeah, that was pretty incredible. I do believe I did shed a tear.
I'm sure.
It's been about 7 years. It's been 10 years. But to get to that point was very difficult, because it wasn't just a matter of doing a simple experiment. We had to build the hardware. We had to build the what's called the consumable. And these are all very complex processes that I actually developed 15 years ago. So I'm leveraging my background. But I had to go to my network and beg, borrow, steal. I literally went into a friend of mine's company and said, I need to do work in that lab, and you're going to let me do this. Do that work. I mean, you just have to be persistent.
Yeah.
And
I can't imagine a stronger motivating factor than your wife.
Yeah, it's motivating, especially when you have little kids.
Yeah.
When we finally got that data, and again, I can't emphasize enough how Arvind and the IndieBio staff pushed us. Literally just, again, we went from scientists and engineers to entrepreneurs, and we push hard. And it took us through the program and then a couple months later to get that data. So what was the feeling like? It was relief. And we were overjoyed, and we had lunch. And then after we got done with lunch, we said, okay, now we got to figure out how to push forward. So getting the data is key. So now we have the data, we have a story, we have the data, we're getting the attention of the right investors.
Talk about the investors, though. They're looking at something that we're trying to see the future, and there's great potential, but you're selling something that may not happen. I mean, it may not work for them in terms of the investment side. How do you resolve that with the investor who wants a 10x return on their money in a certain amount of time?
You have to find the right investor that understands where you are as a company, and understands that it's a technical potential play, that there is a return on investment, but they also understand where they're at in the risk. Right? Still is high risk, but you have to find those investors that are willing to take that risk, because the upside is pretty tremendous. I mean, we entered into IndieBio working in a $1.6 billion market, and we exited in a $63 billion market.
Wow.
Yeah.
Okay, so that changes the discussion. 1.6 billion is not that attractive. A $63 billion market, and we can truly say, look at our tech, look at the other techs, and you can see the potential. So people are seeing that. People that are in this, it's called the molecular profiling space, say, holy CRAP, if you can do that, that is a total game changer.
Is your IP protected?
Yes. Yeah. Our approach is we designed the approach over based on some existing work I did like 15 years ago, and we looked at this whole way of doing it. Of running these experiments and generating the data. We teamed up with probably one of the best attorneys in the Valley. His name is Vern Norville from Wilson Sonsini. Mm-hmm. He looked at it. We did freedom to operate analysis recently, and there weren't any glaring issues, if you will. And we're very confident on the IP side that we have something that's different, that is defendable, if you will.
Right.
Is there a practical application or bridge over to the autoimmune side as well?
There is. We're focused on cancer applications.
For sure, yeah.
But there are definitely applications in Alzheimer's and other diseases.
And still late stage, you think, or is it?
That depends on the disease.
Okay.
Again, I'm hyper focused on cancer.
Yeah, right.
Again, my background, I'm not an oncologist.
Yeah.
I build tools. I'm a
Hashtag
scientist.
not oncologist.
Yeah, there you go. There you go. But what I've done is again, when I got entered into the Stanford visiting scholar program, I immediately just started talking to every oncologist I could find. And there's one in particular, her name is Stephanie Jeffrey. She's a surgical oncologist at the School of Medicine at Stanford. And she's the self-proclaimed queen of liquid biopsy. And what a liquid biopsy is it's a biopsy from the blood, essentially.
Okay.
Right? You take blood and there's floating DNA in the blood. It's called circulating tumor DNA that you can use to look at an individual's cancer. Mm-hmm. And then there's also circulating tumor cells. I mentioned those earlier, right? And so she's a sort of global expert in the area of circulating tumor cell biology and metastatic cancer. So I literally stalked her at a meeting. I found out she was a chair at a meeting. I went to the meeting. I said, I've got to get in bed with her. Yeah. She's the leading expert. And if she tells me this is a dumb idea, then I move on and go get a job and get paid. And so I literally stalked her through the meeting, tapped her on the shoulder, introduced myself and said, this is my idea. And she said, you and I are on the same page. Absolutely, you need to join my lab. So after Ron Davis's lab, I joined her lab for 2 years. Wow. And just continued to network with the experts in the field. Mm-hmm. Right. So again, our company, we're not oncologists and we don't want to hire oncologists. We build tools. We build platforms very well. We're collaborating and teaming up with the experts in the field. Mm-hmm. So we have collaborations set up with Dana Farber, a guy named researcher named Carl Navina, who's doing immuno-oncology and what's called CAR T therapy, and he sees our technology as the only technology that he can see to advance his research. And it's incredible research. And the cool thing about Carl is he's working on my wife's cancer.
Wow.
Yeah, so that's kind of cool. And then Stephanie Jeffrey as well is on board, and then there's some other corporate sponsors. But we've worked hard to build that network of the best in the field. Right. And I query them frequently and say, okay, are we insane? And they say no. Well, we're a little bit insane.
Yes.
You got to be, you got to be insane to do this.
Yeah, most definitely.
To quit your job and pursue something like this.
Yeah. Mm-hmm. You look like you're like, this is kind of crazy.
Yeah.
But you're positive about it. And that's the cool thing. And thank you from all of us in the world for doing this. Where are the experts at that you don't have collected yet then? Are there some like in New York? Are there some in Israel? Like where do you have to collect these experts from?
That's a good question. We would love to go into Asia, and we are talking to people in Asia. We have investment from Asia. We've traveled to Asia several times. We were in mainland China talking to the executives at an organization called Cloud Health 99, which is mainland China's largest sequencing facility, and it's helping to drive their personalized medicine initiative. And Asia is a big market, and we're exploring those opportunities. We're a tiny company going into mainland China right now. Doesn't make a lot of sense, but we're exploring those opportunities. So Asia, Singapore, Malaysia, Japan are areas that we have been looking into. Again, we came into IndieBio and said we're going to do it, and we just got on a plane and set up meetings in Hong Kong and Singapore, mainland China, and it's about having that personal communication. So, or face-to-face is what I'm saying. Mm-hmm. Asia is really where we want to...
You want to expand, land and expand.
Yeah. And in Europe, we've got several people that we're talking to in Europe. But again, that 4-year period when I was in Ron Davis's lab and Stephanie Jeffrey's labs was spent finding the best, cornering them, and saying, "Am I stupid?" Yeah. And so far, none of them said that.
Yeah,
they said
we're not saying it either.
I'm a little insane. I'm a little insane. What I mean by that is, am I on the right track?
Yeah. And actually, it sounds like you're in front of everybody on the track in a lot of ways.
Yeah, they're very enthusiastic, and several of them are bona fide experts in my wife's illness. Mm-hmm. Right. Okay. And some of them I sought out because they were experts. And in the case of Carl Navina, I did not know he was an expert. I was just introduced to him through, we were doing some customer interviews, a course called Lean Launchpad out of UCSF where you test your product concept in the market before you go build it, right? And so we were interviewing everybody we could find. And one individual at Dana-Farber I was referred to by a friend. He said, you should talk to Carl Navina. So I talked to him, told my wife's illness. He said, I'm an expert in that. And that's just how it happened. Now he's a very good friend of mine, very supportive, and it's just things just happen. Yeah.
Yeah. I think that the process that you've gone through leading up to this is a very classic entrepreneurial story where you just have to hustle, knock on doors, persevere, and networking is so key to, having success.
Yeah. And you also have to be humble and don't take anything personally.
Right. Yeah.
Yeah. Could the younger you have pulled this off? Like, did you have too much ego question. 10 years ago?
Yeah.
I don't think I've had a big ego. I think I have a bigger ego now by design.
Yeah.
You have to have a little bit of a swagger, I think.
Right.
But no, I mean, I don't know. I don't know if I'd have the confidence when I was younger to just basically quit my job and just go pursue people, like these potential Nobel Prize winning people.
Yeah.
I met George Church.
Hashtag still not an oncologist, right?
Yeah. But for instance,
oncology.
in mainland China, George Church was on the SAB of this company called Cloud Health 99. I was there for the grand opening. A friend of mine's the CTO and invited us over. And had I not had this drive, I mean, I've got to do this, I never would have approached him and said, hi, Dr. Church, my name is John Butler. This is a technology I'm building. What do you think? Now I do it because, these luminaries are just people.
Right.
And they're scientists. And so a younger me, I don't know. That's a good question.
Yeah.
I mean, the circumstances, I have to do it. It's not.
You don't have a choice now.
Yeah. Yeah. So, and my wife is 100% supportive and obviously, and it's been an interesting ride.
Yeah. What are some of the challenges you faced along the way? Like anything in particular that was a particularly tough moment that you had to?
A lot of the challenges are in getting the right quote unquote advisors. So a lot of people want to come in and present themselves in a certain way. And then you bring them in, you trust them, and perhaps they're not delivering on what they said they would deliver on. I think most of the challenges are in continuing to justify pursuing this in light of raising 2 children and your wife. And my wife, again, she's amazing. She's taken the burden of supporting the family while we've been going down this path.
Wow.
It's hard to say. I think the other challenges are at one point you're stopped and you want to continue to move forward and you don't have the funds to do it. Yeah, it's just hard to sort of the challenges I can come up with.
Right.
Indie Bio was a challenge because, again, these guys forced you to change the way you approach the world, right? Being shameless a little bit and figuring out how to sell. I sold my first instrument 2 weeks into the program because I asked Ryan Bethencourt to show me how to sell. And he said, I'm going to do a training. And he said, well, who would you call right now to sell an instrument? And I said, Carl Navina.
Yeah.
And he said, okay, call him right now, put it on speakerphone, everybody be quiet, and sell him an instrument. And I went, holy crap.
Okay.
Luckily, he's a good friend, though.
Well, at the time, he wasn't that good.
Okay.
I mean, we've become friends, but we were talking about collaboration and so forth, and so I just did it. And then Ryan said, that's how you sell.
And he said yes.
Yeah.
That's how you
LOI. So, yeah.
You closed the sale, yeah.
Yeah,
nice
but again, I think again, back to the challenge thing. Things were challenging, but there was a goal here, right? We had to go down this path. I was working with some amazing people. I think once we raised, I think it was 675 coming into Indie Bio, that's when it became challenging.
Yeah.
Because now we have to produce, we have to get the data, and all the data we had was conceptual.
Right.
Right. And so that's a lot of ups and downs in that.
Talk a little bit, go back a little bit further, and because you've cast the die, right? You threw your hand, there go those dice, right?
Yeah.
The moment before that, as you were, you've got the news from your wife, you realize this is a life-changing problem, and you've got the dice in your hand. Talk about that. Like, what were you thinking? Because it's bigger than just saying, let's try this.
Yeah.
Like, talk about that. How was that?
So the first thing was stop challenging her oncologists on everything they said. I was actually excused from one of her appointments by her because I was right and the doctor was wrong, but I don't want to go into that. Initially the first thing you think of is how do I make sure my wife's well?
Yeah.
How my kids, because we had a 6-month-old and a 3-year-old when she was diagnosed. She was 32. The first thing I thought of was how do I get involved, say, in a nonprofit setting? Could I become a patient's advocate? Right? Because the first thing was I want to push the research. as hard as I can.
Yeah.
So I thought I'll become a patient's advocate. Well, there's a bit of a problem with that, and I'm not very political.
Okay.
I couldn't figure out that avenue. And then again, it came down to what are our core competencies. So that period after she was diagnosed and then we sort of got started on this was about a 4-year period, right? I was at this company, Pacific Biosciences, that had probably one of the most amazing scientific and engineering staffs of any company I'd ever been in. And I was just sort of talking through this. How do we do this? How do we advance cancer research? And so it was very much of a lunch every quarter or something like that.
Right.
And then again, you have that aha moment where you say, wait a second, I can do this with
This
technology.
works.
Yeah.
What was that? So what was that aha moment?
I think the aha moment was when we decided on the approach. We looked at what we were delivering to the customer. Again, we have to convince the biologists and the cell biologists. And then when I stalked Stephanie Jeffrey and said, this is what I want to do. And she said, you and I are getting together. And she's become a mentor and a dear friend to me. And that's when I went, okay, we're going down this path because I've got the support of the best in the world. But she was realistic and said, you're way ahead of the curve. That's a hard problem to solve. Right? So we want to solve that problem eventually, and we will get there. It's not the first problem we're going to solve because it's still fairly difficult, but we will get there.
How hard has it been? In my field, I used to be a spy. I worked in Iraq and Afghanistan, and I did a lot of stuff with culture. I've got to say that so you can understand my question. And a lot of the things I did were way far ahead of my peers because they want to go in and do tactical things and shoot people. And I'm like, hey, if the people don't believe in what you're doing, then it doesn't help.
Yeah.
So I was always miles ahead of what they were doing because I was on the ground actually doing it. And I see that you have the same kind of thing, like you're actually looking at this data and trying to figure it out. But when I look back, I'm not leading anybody because I'm so far afield. Yeah. How do you deal with that?
Yeah, I like to say, at one point in my life, I'd like to no longer be on the bleeding edge.
Right.
I'd like to know what the edge is like because I probably will get paid consistently and not constantly wake up going, what the heck am I going to do today?
Who's this guy? That guy over there waving his arm.
Yeah, so it is tough to do that. It's just in our nature. It's in, again, myself and our CTO's blood. We're always trying to push. That's what we've been trained to do all our lives, right? So when you're building products, you're always looking at, okay, here, you launched that product. Now what's the next one that's going to keep you ahead? And we have to look at this from two perspectives. One, building a business, because we have to build a business, and how do we stay ahead? And also, how do we help patients? Because our goal as a company is to help patients. And we know with our ideas, if we can do what we say we can do, we can help patients, and there's a ton of patients we can help. So from a business side, it makes sense. But understanding that market and selling that is not as easy as you'd think, because what we're talking about is a cancer agnostic test, and the world thinks of things in terms of breast, colon, prostate, and cancer types.
Yeah.
But that people are changing that thinking. But we were proposing this again 7 years ago.
Hey everybody, Pete A. Turner, co-host of Popping the Bubble. This episode is sponsored by us over here at Lions Rock Productions. What do? Content management strategy. We'll help you create podcasts, develop strategies, consult on what you've got. If you want to take a look at content, we would love to have an opportunity to do that for you. Now let's get back to Popping the Bubble.
So you're saying your technology, it doesn't matter what kind of cancer the person has, it will react the same.
It'll give you information. It'll give you information on how that particular patient might respond to a specific drug.
I see.
That's the goal. That's where we want to go. Again, it's still a long road to get there, but we're always, that's how we think. How do we get to that point? It's what's called personalized medicine.
Right.
Yeah.
Right.
Yeah. So we don't care about what type of cancer you have. We care what Stephanie's cancer is, and we help her.
We've learned a lot about this in our shows, that this personalization of whatever it is going to be, whether it's caring for your skin or caring for your cancer. How do you take such a big, crazy goal and tactically approach it, like every day? Like, how often do you have a breakthrough? What leads to those breakthroughs? All these day-to-day work things. How do you do all that?
So what leads to a breakthrough? Every experiment we do is a breakthrough. Again, our cell biology staff, her name's Dr. Suhani Thakur and Katie Koenigsfeld, are the two that are doing the experiments.
Yeah.
And every time they do an experiment, we learn something. There's also our bioinformatics guy, his name's Aaron Arvey. Every day when they get a new data set, it's a breakthrough. It's an understanding of the technology and our benefit, because we truly, with the data we have, we outperform current comparable technologies for the most part. And it's giving us more information on we're getting more confidence in what we think we can do. This is what I'm trying to say. Validation, yeah. I don't know if I answered the question, but again, it's every day is a new something, right? And since this is our first company, everything is new, and I wake up every morning going, okay, now how do I deal with this?
Yeah, so that's
Okay.
the addendum to this question.
Yeah.
What keeps you up at night?
Everything.
Not good enough. Give me some details.
What keeps me up at night? Money.
Okay.
Always worried about money. I have to say that because my good friend Arvind Gupta would get mad if he didn't think I was always focused on getting money for the company, because my job. Data, whether or not it's taking us too long and we can keep the collaborations together, because our current collaborations are on paper and we want to get those started. So you're always thinking about whether or not those individuals are going to stay on board, or if they're going to get funding from somewhere else and be able to continue to talk about help you push the technology forward. I worry about supply of materials I mentioned Katie, Suhani, and Aaron. When am I ever going to get paid? Because currently the executives aren't getting paid, because the founders aren't, because we're just in this position where we said we're not going to get paid. It's by choice.
Yeah.
We want to get to that point. Will we make it through due diligence? Because if we don't get money, we die.
Right.
Yeah.
And so
you guys are currely in a funding cycle then.
Yeah.
You guys got some initial funding from IndieBio and some other investors.
Yeah, so Indy came in, we've raised to date about 1.2 million. We use those funds to build our instrument.
Right.
We built 2 instruments.
Are those manufactured here in the U.S. or?
Yeah, they're in the company, so they're prototypes.
Right.
And we use that to get to our current data set, and again to build these 2 amazing pieces of equipment that Bidhan has built. So Bidhan comes from Intel, and he brings in a high-tech engineering perspective to a biotech application. And the stuff that he does is just amazing. I've never seen engineering like this in my entire life. So we've raised about 1.2 million. And now we're doing our equity round, and we've been talking to various investment groups, family offices, venture capitalists. And
you're looking to raise how much more?
Looking to raise $5 million. $5 million gets us through our beta program. So our beta program is actually a revenue-generating program where we have 3 or 4 key opinion leaders. We already have 3 set. We have a 4th that we can sign on. But that beta program is where we work closely with them with our prototypes, or actually what's called a beta unit, which they purchase, and they also purchase the consumables. So it's a revenue-generating operation or project, and we just validate our technology in their hands. So we'll be looking at preclinical samples, we'll be looking at clinical samples, and from the company's perspective, it's really a marketing campaign. Because the way you market these products is you go to conferences, you publish, you present. So those funds will pay for that beta program, building the beta units, hiring staff, building the marketing program, and at the end of that, we're at a point where we're ready to start production, meaning that we've got our entire supply chain figured out of materials. We have all of our vendors in place. We have our third-party contractors in place, all the documentation, and we just need to do a raise to build our inventory.
Right. You've got everything projected, so you know what it's going to take to scale.
Yeah.
And
exactly.
now, and is there any involvement from the FDA or other
we are
organizations?
So we have conversations with the FDA. I have a friend at the FDA we've been talking to for several years, and the way you sort of approach the FDA is to get them involved early. We are unclear as to whether or not it's an FDA regulated product.
Sure, I can
and
see
whether
that.
or not, and it's kind of hard when people ask me, do you need to be FDA regulated? At this stage of the company, at this stage of the product, I can't make that statement.
Who else might play in there? Ag or what was? I mean, it's always weird who plays in these spaces.
So we've actually been contacted by Monsanto. And we're chatting with them. I actually early in my career was in ag.
Yeah.
And so there are some.
Isn't it funny how ag always comes in like this? It's like you wouldn't expect it. That's why I was asking. They're odd players in this space.
Yeah.
I actually had Monsanto on my other show one time. It was really interesting talking to those guys because they do world-class science they just piss everybody off in the process, you know?
Mm-hmm.
Yeah, I was actually I was telling somebody yesterday, I was at the conference where they debuted the Roundup resistant soybeans. This
really?
was back in 1989,
Wow.
so I'm dating myself.
Yeah.
But that's when they debuted it, and I was sitting next to scientists that I was in awe of, and I would never talk to him. And I looked at him and I said, looks like we all need to find a new job. Because I didn't work at Monsanto. It was impressive. It's impressive. I mean, it was startling.
So as far as the regulatory thing, it's TBD. We're going to find that out.
So what we're doing is we're making sure that we have the systems in place so that if we have to go down the regulated path, the audit process is simple, and that's if you prepare early in a company, your business systems or quality management systems with that in mind.
Yeah.
Getting there is not as difficult as you think. It's where you get tripped up is if you don't plan for that, and then 3 years later you decide to go down the FDA route. You have to do sort of retrospective analysis and put all those documents together. But that's how Bidhan and I do business. We've done that our entire lives. We've had these systems in place.
Is anybody standing up and saying, we think we're the ones that are going to regulate you? Like, is Department of State saying, we think it's going to be us?
No, we're too small.
So no one's really claiming you yet.
Yeah, I mean, we think the path, there's a couple of different paths to building the business. One is we'll go out to the research market and sell tools and consumables, sort of a classic type of approach, and then generate data to see if we're going to go regulated. The other path is to partner up with some of these reference labs and develop tests with them. Sense. And that's something we're exploring as well. And that's actually becoming more and more attractive because, again, our core competency, what we do really, really well, I'm not afraid to say we're kind of world-class at this, is building these platforms extremely well, extremely robust, and they work every time. That's our core competency. We want to partner with the experts and the people that can take it from there and, again, do what we want to do, which is help cancer patients.
Yeah.
All of them.
I mean, it seems like there's definitely a lot of attention in this market and specifically trying to solve or find cures, treatments for cancer. Are there other technologies that you've seen or other approaches that you've seen that you admire or that you think could also work? Or is it just our way is the only way?
No. There's some technologies out of MIT. Chris Love, who's actually on our SAB, that are quite attractive. Very cool tech. I mean, there's on the techie side, the technologies are. If I'm being my technical self, I'm in awe of all these technologies that are out there because there's some incredible ones. 10x Genomics has one where they can sort thousands of cells and do look at a large number of cells very quickly. Very cool technology. A company called Fluidigm they've been around for quite a while with a microfluidics approach that's very cool. We bring in a dimension that most of them can't bring in, which is we can associate what the cell's doing, the whole cell, with its genetic information, right? And we can do that on large numbers of cells. And what that and it's being able to associate again, it's called the phenotype, how the cell's responding to its environment with the genotype. That allows you to answer questions, why is the cell responding different? So in cancer's very what's called heterogeneous. You don't consider each cancer cell is different, right? What we want to do is we want to take a patient's cancer cells and we want to treat them with a drug, if you will. We want to measure how each individual cell is responding. We want to separate them out into responders, cells that are responding to the drug if not dead, and those that are not responding. And then we want to look at what's called the transcriptome, or the RNA sequence and expression of each one of those cells to try and figure out why didn't this cell respond. And within that information from that individual cell or those non-responsive cells, we might be able to find markers for existing drugs, other drugs. So now you have Stephanie, I'll use her, she's my wife, who 90% of her cells respond to this particular drug. The ones that don't respond, we found a marker that shows they respond to this drug. Now I'm going to give her 2 drugs.
Oh, okay.
And then you continue to monitor that over time. And that's kind of, that's being able to associate that whole cell response to that genetic information at a single cell resolution. That's really kind of cool. Yeah.
It's all about the RNA.
It's about, and we actually have a second application that we're working on, which is where we're going to take a solid core, a tissue, tumor biopsy that you get as a standard test from a histopathologist. So when you get cancer and they resect the tumor, they take a section. It's called a microtome section. It's about a little less than the thickness of a cell. And they put that onto a microscope slide. And the pathologist will stain it. It's called H&E staining. And they'll stain it with specific markers that allows the histopathologist to look at that tissue and tell you the aggressiveness of your cancer and whether or not you have specific markers for drugs or something like that. Well, the technology we are developing, our second application, we can interrogate cells within that tissue section, and not a couple of cells. We can look at hundreds of cells on that tissue section and look at their sequence data.
Wow.
Right?
Yeah.
And so now what's cool about this is we can look at the tumor section, get that information, but we can also associate what's called the spatial data. So where is this cell relative to other cells around it, and how is it interacting with those cells? It's called the tumor microenvironment. A good friend of mine I was talking to yesterday about the application, understands more of the drug response, said that people underestimate influence, if you will, that this tumor microenvironment has on drug response, and people are appreciating that and trying to figure out how to study the microenvironment. Yeah. So this technology or this application allows you to essentially say, there's my T cell, there's the epithelial cells around it, and I can now look at how they're interacting relative to the spatial context of the tumor. Sorry if that's a lot.
No, I mean it's you're blowing my mind.
Yeah. And so again, we're techie guys.
Yeah.
You know, this is Indie Bio. One day somebody asked us, could you do your single cell stuff on a tissue? And we said, well, I don't know. And then we started looking at the market. We started looking at the potential. And that's what moved us from a $1.6 billion market to a $63 billion market, that question. Mm-hmm. And so, you know, we looked at it from a technical perspective. Could we open this up? We weren't looking at so much. We were understanding the data as we're going along, but we figured out a way to do this. And so that is something that we're extremely excited about, and people are really excited about that because it really, again, accelerates the process. You get 2 to 3 orders of magnitude more data per tissue than with existing technologies, and it's automated. Mm-hmm. So you can drop the cost, you can get a lot more data. We're pursuing that as well, as much as we can with the minimal resources.
When we come to check in on you in a year, you're obviously not here at Indie Bio anymore. Are you at J Labs? Are you in Stanford? Where are you guys sitting?
We're at, so in a year, we'll have our own lab.
Yeah.
But is that going to be here? Where are you going to be?
Oh, in the Bay Area.
Okay, so you're going to stay local. You think, okay.
Yeah, I mean, I was born and raised in the Bay Area. Mm-hmm. Right. I live in the house that I grew up in.
Wow.
Oh, wow.
In San Francisco?
In San Jose, in Willow Glen.
Okay.
Yeah, I bought the house from my father. Prop 13 tax laws are very favorable.
Yeah.
I can explain that another time. But
Jarvis can.
Yeah, but no, I mean, this is where I expect to be. We'll have our own lab, and we'll be deep into our beta testing and getting tons of really good data and presenting at conferences. And actually, in a year's time, I will be probably 3 or 4 months into working on our Series A funding.
How cool is that?
We'll see. Actually, it is. Fundraising is tough. It's a grind. You just keep grinding, and it's just a matter of finding the right investor.
But you're fundraising about something that's going to change humanity. You know, it's one thing to have a widget, you know, that's going to make doing laundry easier or whatever, and that's cool. but you're doing something that, as an investor, you go, yeah, you know what, I got some money to help you guys figure out cancer. So it's
yeah, and again, it's finding the right investor, because you can get money, and money's great.
Yeah.
But at this stage, you want somebody that's going to help you strategically and open doors,
right,
and support you,
and be in it for the long haul as a partner versus just wanting to have the quick exit. Yeah. We're not in this for a quick exit. But you know what? Again, the people have asked me, what's your exit strategy? And I don't know what that is. I can't predict that now. Mm-hmm. The exit strategy is whatever's right for the business.
You're far enough ahead of everybody. You just keep on going the direction you're going. You know?
Yeah, like I said, it'd be nice to be on the edge at one point, but I probably wouldn't get bored. Yeah. And my wife would probably say,
get out of the house and go get on the edge.
Go get on the bleeding edge, because you got to bleed.
Yes. Mm-hmm. That's what you have to do as an entrepreneur, for sure.
Yeah.
Mm-hmm.
Yeah.
So the best way to get a hold of you guys and find out about you is I know on Twitter you're at QuantumCyte, but what's the best place to find out more about what you're doing?
The best place is at QuantumCyte, because I am posting frequently. I typically do that through my LinkedIn account.
Great.
Onto Twitter. My email address is jbutler@quantumcyte.com. That's the best way to get a hold of me.
And you're looking for potential investors, other collaborators?
We are looking for collaborators. We are looking for investors, people that would either want to lead or follow. I think we're close to getting a lead, but people that would want to follow. And people that are, just generally interested in learning more about what we're doing. Partnerships are always great. Again, we're talking to companies about partnerships around integrating into workflows and so forth. I mean, that's really what we're looking for now. Once we get our equity round, we'll be looking for cell and molecular biologists, engineers, business development people, and so forth.
Yeah.
We're fortunate in that we actually have quite a few people already signed up to join once we get funding. But I'm always looking for talent, because people are what make you successful.
Absolutely.
And every time I talk to somebody, I've explained that whenever I talk to somebody, I'm always thinking, how could you help me out?
Yeah.
How could you join?
That's a good way to do it. You have to.
Yeah.
You're turning over rocks. You have to.
Yes, I'm now a CEO. I'm no longer a scientist. Every once in a while, I get to be a scientist, but then Suhani and Katie sort of put me on my place and say, that was a very nice idea. Let us continue on, because it's a really complex product. You have to be really good, and we've got some really, really good scientists.
Yeah, let those guys do their job.
They just get it done. I mean, I'm floored at what they have figured out with this technology. It's really cool.
That's exciting. Well, it's been a great conversation. Is there anything that we didn't talk about that you maybe wanted to mention? Super fascinating. And I love your founding story and journey. I mean, it's just so compelling, and we are going to be looking forward to watching QuantumCyte as you progress.
We're looking forward to it too. It's going to be it's been fun. I always joke, I always say it's a little bit stressful, but it's fun. We're doing it for the right cause. It's not like I wake up in the morning and I'm like, God, I don't want to go. It's like, we got to go.
Absolutely.
And the entire team is there too.